Good morning, grade 10. Today we are going to continue section 5 from chapter 8 as a revision for the second exam, translation. We said the translation is converting an mRNA message into a polypeptide chain, which is a protein. We learned that amino acids are coded by mRNA base sequence, as you see here. On the mRNA, we have codons. Each codon contains 3 nucleotides, and each codon is specific for a certain amino acid. So codon is a sequence of 3 nucleotides that code for an amino acid. This segment is on the mRNA. It must be translated, so the codon will be translated to an amino acid, and the ribosome will be able to join these amino acids and form the proteins. As you see here in this table, you can observe that each amino acid can be coded by more than one codon. Like phenylalanine has 2 codons which code for it. Okay, now the genetic code matches each codon to its amino acid or function. You have 3 stop codons, 1 start codon. Okay, the start codon should be methionine for translation to happen. Now referring to the chart, which amino acid would be encoded by the mRNA codon CGA? If we refer to the chart back, you can observe that CGA, where is CGA? So here you have, it codes for arginine. Can you see? So this CGA codes for arginine amino acid. Now, change in the order in which codons are read changes the resulting protein. And regardless of the organism, codons code for the same amino acid. That means humans, animals, bacteria, plants, each codon codes for the same amino acid, whatever the organism is. Now these amino acids are going to be linked to become a protein. How this will happen? For making the protein, there are 3 parts which are important. The first one is the tRNA. tRNA is an RNA molecule which is responsible to carry the amino acids, transfer amino acids from the cytoplasm and carry it according to the anticodon it has, which join the complementary codon on the mRNA. So the anticodon is a set of 3 nucleotides that is complementary to an mRNA codon. Now, an anticodon is carried by a tRNA, and this codes for a certain amino acid. So look at the tRNA, you have at the bottom of the tRNA you have the anticodon which will be joined to the mRNA codon, and the upper part is the amino acid which is in which this tRNA bring from the cytoplasm according to its anticodon. Now ribosomes is the second part that is responsible for the translation process, and ribosomes consist of 2 subunits, large and small. Now the large subunit will bind to the tRNA and the small subunit will bind to the mRNA. Okay? Now let's look how this works. For translation to begin, tRNA binds to a start codon and signals the ribosome to assemble. Now tRNA will bind to start codon, which is methionine, okay? And now the ribosome have a signal to start assembling the protein. Now the complementary tRNA molecule binds to the exposed codon, okay, bringing its amino acid close to the first amino acid. As you see here, can you see the tRNA? The exposed codon in the first site attracts a complementary tRNA. This exposed codon on the mRNA will attract the anticodon from the tRNA, and the tRNA bear the amino acid from the cytoplasm. The first one is the methionine. Now the tRNA anticodon pairs with the mRNA codon, bringing it very close to the other tRNA. Now the ribosome forms peptide bonds between these two amino acids, okay, and it breaks the bond between the first tRNA and its amino acid, and now the tRNA will be free. All right. Now the empty tRNA molecule exists in the ribosome. It will exist out. A complementary tRNA molecule bind to the next exposed codon. So once the stop codon is reached, now this ribosome will keep moving a length of one codon. When it reaches the stop codon, the ribosome releases the protein, because here the amino acids are bound together in a polypeptide chain. The protein is ready. This protein will be released. Okay, this is the main important parts of the translation. You should know the parts, which are the ribosomes, tRNA, mRNA, and you have to know how they work together to synthesize the protein. All right, now let's go to the next section, which is the gene regulation, expression, and regulation. Now we learned that the transcription should be regulated. Why? Because transcription for protein synthesis happened according to the need of the cell. If the cell needs, for example, a certain enzyme, if the body needs a certain enzyme, so this enzyme will be synthesized by expression on the DNA. Okay, this expression on the DNA will be transcribed and it should be regulated. So transcription cannot be happen in a proper way if it's not regulated. It should be regulated according to the need of the cell. So gene expression is carefully regulated in both prokaryotic and eukaryotic cells. In prokaryotic cells, it's much more easier than prokaryotic cells. The cells turn genes on and off by controlling transcription. Okay? Now, in prokaryotic cells, we have promoter, which is a DNA segment. This promoter is important. Why? To allow a gene to be transcribed, starting to let the gene start to transcribe and to specify a location from where to start. An operator is the part that turns on and off the gene. Now, in the prokaryotic cells, there is operons, specific operons, and which includes promoter, operator, and the gene code for all proteins needed. Okay, so operons are most common in prokaryotics. If we take one example of operon, there is an operon which is specific for lactase protein. It's called LAC operon. Now, the lactase protein enzyme needed to break down lactose. If the bacteria consume lactose, it needs to break it down. So it needs the enzyme lactase. And this enzyme to be synthesized, it must be transcribed, and this transcription should be regulated. Okay? So LAC operon was one of the first examples of gene regulation to be discovered. And it has 3 genes that code for the enzyme that break down lactose. Now, if you look here, you can see the LAC operon consists of promoter, operator, repressor, and genes. Hala, if the repressor is binding to operator, this will block the RNA polymerase from transcription. Okay? So the LAC operon is off when lactose is not present. If lactose is present, the repressor will bind to the lactose and the RNA polymerase will be free to start the transcription. Now, in eukaryotic cells, we learned that we have different sets of genes are expressed in different types of cells, okay? And this is controlled by something called regulatory DNA sequences, which composed of enhancer, promoter, okay? And we have the RNA polymerase. it will start to work when these enhancer and promoter come close together. This happened actually by binding of transcription factors to enhancer. Then the DNA strand will bend to make the promoter enhancer close together, and RNA here will start transcription for the gene. Transcription is controlled by regulatory DNA sequences and protein transcription factors. And most of eukaryotes have a TA box, as you see here, promoter. Enhancers and silencers speed up or slow down the rate of transcription. And each gene has a unique combination of regulatory sequences. Now, after transcription of RNA happened, RNA should go through processing, which is an important part of gene regulation in eukaryotes. And processing includes three major steps. First, the introns parts of the mRNA will be removed. The second step is a cap will be added, and the last one is the tail for the mRNA. So exons are nucleotide segments that code for parts of the protein, while introns are nucleotide segments that intervene or occur between exons. They have non-coding parts. mRNA processing includes these three major steps. So when the mRNA is ready, now it can leave the nucleus out to the cytoplasm. So processing of mRNA will happen in the nucleus. Introns are removed and exons are spliced together, attached together. A cap is added and a tail is added. Thank you for listening and take care.